So the topic for today's environmental microbiology project is metagenomics, and my name is Preet Kaur,
And my name is Kuljinder Singh Sidhu.
and we will represent this topic. So moving on, we will start with the introduction. The study of metagenome is called the metagenomics. Now the question arises, what is the metagenome? The metagenome is a collection of genomic sequences of various microbes obtained from the environmental sample, and the sample could be soil, water, or sludge, or many more examples are also there. So basically, it is a collection of genes. The purpose the purpose of metagenomics is to obtain the information on the microbial diversity and ecology of a specific environment. So specific environment being the sample, and we are just trying to find out what kind of microbes are living in that sample. So metagenomics help with that.
Well, in metagenomics, we sequence whole genome regardless of the samples collected from which environment. Moreover, it is used in large-scale genomic applications as it helps us in study of taxonomic and functional composition of microbial communities from different environments. The other application of this technique is that it's used for the identification of unculturable microorganisms, like which cannot be cultured in the laboratory.
So moving on to the next slide, it says why metagenomics. This slide tells us what is the need of metagenomics. The conventional methods of studying microbiomes have some limitations. That is, vast majority of microbes cannot be cultured by plating due to many factors. As we all know, there are some organisms which we find really difficult to plate and really difficult to culture. So metagenomics help us overcome that problem. Current technology for metagenomic analysis allows us to completely bypass the culturing step and instead extract nucleic acid directly from a sample. So if a sample or if any kind of microbe is present in our sample, with the help of metagenomics, we will just extract its DNA and we will know whether that organism was present there or not. We don't need to culture it. Along with that, it helps us answer the following questions. Number one, who is there? The genetic analysis tells us what is the population of archaea, bacteria, protozoa, and any kind of organism that is present there. Its DNA will be tested, and we will know if the organism is present in our sample or not. The second question it answers is what they are doing. The analysis of the genetic products, that is enzymes, proteins, anything formed from its transcription and translation of DNA and RNA, it will tell us which species is a dominant species, an indication of its biochemical processes.
Next, the experimental plan is that first of all we get a sample from the particular site, then we fractionate the sample as any pre requirements for sample preparation. Then we extract the DNA from the kits available in the market. Then we go for DNA sequencing. Then we, after sequencing, we assemble the DNAs in libraries like complementary genomic DNA libraries. Then we perform a statistical analysis with the help of bioinformatics. Then we store the data and share as per the need.
So now we will learn about the types of metagenomic processes. So basically there are two types of metagenomic processes known as targeted and shotgun. In targeted metagenomics, only certain conserved regions are amplified using the PCR primers and sequence. For example, we will choose a specific kind of RNA, for example 16S RNA, and we will only amplify that. So if there are 100 species present in the soil sample we have taken, our process will only amplify 16S ribosomes of all of those species. This particular type of metagenomics helps us in identification of different groups of organisms. The second type of metagenomics is shotgun metagenomics. It is a non-discriminant method that sequences everything in that sample. Irrespective of the desired gene we are looking for, it will just sequence everything. It will give us a lot of data and help us quantify organisms even down to the species level.
In the target sequencing, we can perform 16S rRNA sequencing, which involves the collection of environment sample. Then we extract the genomic DNA from that sample. Then we amplify the extracted DNA with the help of PCR. Then we assemble all the sequences in the genomic or complement DNA libraries. Then these sequences help us in the identification of phylogenicity of the desired microbes.
So moving on to the next type of metagenomics, that is shotgun sequencing. The first step is to cut the DNA into overlapping fragments short enough for sequencing. So the DNA is cut into short fragments for the software to be able to amplify it and read it properly. Then these small fragments are cloned in plasmid or phage vectors. Later on, each of the fragments are sequenced and they are put into order using a computer software. So this way we have the whole genome sequenced of multiple organisms present in the same sample.
Metagenomics is an upcoming field which has various applications in different sectors. Now we will discuss one by one. The first one is it helps in monitoring and optimizing recirculation of agriculture systems. Second, it helps in monitoring pollution, like it helps in analyzing the different bacteria which live in different uh, pollutants. The third one is it helps in bioprospecting, means it helps in hunting for novel enzymes and compounds, like discovery of the new enzymes or pharmaceutical enzymes, which helps in different biotechnology techniques. The fourth one is it helps in maintaining the function and ecology of the oceans. Then conversion of biomass. It also helps in development of functional feed for the animals or other creatures. It also help in disease surveillance and treatment, as we can analyze the microbes which cause different diseases and we can develop various medications for them. The last one is it improves the yield by knowing the host and microbe interaction and other various fields, various applications which can be developed in upcoming time.
So diving deep into the analysis of applications of metagenomics, let us know in detail about environmental remediation. Metagenomics can help us understand how different microbes cope with toxins in the environment and how we can potentially use them to recover from pollution. If we take a sample from a really polluted area and perform metagenomic analysis on it, we might be able to know what kind of microbes are thriving in that toxin rich environment and how they are able to do that. So, with knowledge of that, we can potentially use their biochemical processes or the products to recover from the pollution. Number two is agriculture. Metagenomics can really help us understand the relationship between microbes and plants. We can know about members in nutrient cycling and various diseases. We still know about the relationship between fungus and plant roots or different kind of bacteria that help in nitrogen fixation. Metagenomics can help us further that knowledge, and we might be able to know which bacteria or which fungus, which other kinds of microorganisms are helping plants and which of them are causing diseases. Number three is infectious disease diagnosis. Conventional methods involve taking the sample from the human body and culturing it and performing biochemical reactions in order to identify the organism, which is time consuming and not always we are able to grow that organism and therefore that disease goes undiagnosed. For example, many cases of encephalitis go undiagnosed despite extensive laboratory analysis. Newer technologies like metagenomics can help with diagnosis because in metagenomics we are just performing genomic sequences of everything that is in the sample. Human cells and bacterial genomics can easily be differentiated using metagenomics. Number 4 is biofuel production and other biotechnology applications. Certain enzymes are required to convert biomass into biofuel. Metagenomics can help us understand which microbes produce such enzymes and which can potentially be used for production.